Hey everyone, we're Dr. Daylon James and Dr. Arun Sharma. We're the hosts of the STEMCELL podcast, and we'd like to welcome you all to day one of the 2020 ISSCR virtual meeting, the first of its kind. The meeting is taking place entirely online this year, so we thought this would be a good opportunity to get outside of our comfort zones and do something a little bit different. Throughout the meeting, we'll be publishing daily videos. It's only a few days, so bear with us. They're going to be just like this one. We're going to be discussing the most interesting research presented at ISSCR 2020 on that day. Since the first day of the meeting was mostly workshops, today we're going to talk about some of the research that we're most excited to hear about later in the week. But before we get to that, we have a message from Arun. Arun, if you will.
So if you're working with human pluripotent stem cells, and you probably are if you're watching us right now, or considering it, you'll probably want to watch the focus session with the Coordinates group. Coordinates? Stem Cell Technologies will be giving a sneak peek at recent insights and trends in pluripotent cell research. So join live starting at noon Eastern Time, that's Eastern Time US, for a Q&A. And you can, of course, find the full schedule for live sessions at STEMCELL's virtual booth and at ISSCR on their website.
All right, with that done and our inaugural video episode underway, Arun, why don't you start this one, the first, by telling us what you're excited about this at the meeting?
Yeah, it's a whole new world, right? You know, we can't go out there and, see people in person, give a hug or a handshake that, we normally want to friends when we're at these conferences, right? But we got to take it in full stride, right? But there's so much good science that's still happening right now, right? I'm actually really interested not only in, of course, all the organoid stuff that's inevitably going to be talked about, but one actually unique aspect of the conference this year is there's a COVID-19 centric session, actually. And of course, some of the biggest names in stem cell biology are going to be on that particular panel. But one particular name actually really stands out, and that's actually Dr. Anthony Fauci, who's, of course, super well known when it comes to all things COVID-19, right? He's one of our nation's leaders when it comes to discussing COVID-19 and thinking about strategies to treat that. So that's one session that I'm really excited about. And of course, there are a ton of other big time sessions with speakers like Sek Kathiresan, who was formerly at the Broad Institute, and now he's at Verve Therapeutics. So I'm interested in listening to him. Of course, interested in listening to Shin Yamanaka, the godfather of iPSCs himself. That's always a fun talk to listen to. So tons and tons of amazing stuff to listen to. And of course, I want to give a shout out to all the trainees out there. I know you guys aren't able to do your poster sessions in person, but those poster sessions are still online. And I'm encouraging everybody to stop and give some love to the trainees, right? Because I know you will, Daylon.
I always do. I love the trainees. I need trainees at the caliber that are floating around at this meeting. It's going to be a good one. I got some highlights here. I'm going to go through them pretty quickly. One, there's this session on SciComm tomorrow. I want to get into that because that's what we do, Arun. We are science communicators, if you didn't know. Also some ethics things they're going to do tomorrow. There's the early career panel. Check in on that. Thursday, the plenary, I didn't mention the presidential plenary. Also highlight there is Susan Solomon getting the Public Service Award. I'm a former NISA fellow, so that means a lot to me. I think she deserves it. A cancer survivor herself, came back from the brink. So big up to her. She deserves it. Also in the second plenary is the Young Investigator Award. I'm going to check that out, going to Alon Klein. There's this interspecies chimera. There's a whole bunch of stuff going on with there. Martin Perez is going to introduce, and it's going to end with Jun Wu, who we had on the show almost a couple years ago now, when he was on the chimera with the pigs. So I can't wait to see what he's got to say. Also, Benoit Bruno, we're going to talk to him on the podcast, so we got to check in with him in the plenary. The third plenary, also Elaine Fuchs, one of my idols from my alma mater, Rockefeller. Then the concurrence. Oh my God, the concurrence, they're exhausting. I think the good thing, Arun, about the concurrence, though, in the virtual format, I mean, we're going to get back to what the pros and cons of the virtual format are. But I think the one thing that's totally positive is that you can bounce between concurrence like a butterfly, whereas before you might work up a sweat doing that. What do you think about that?
I was actually, that's that's something that's super easy. You just click a button and you can go back and forth from one concurrent to another, as opposed to, leaving a particular room, running down the hallway and going into another room, right? So it makes things very straightforward. And I think another thing that, this whole digital format is really facilitating is when it comes to the trainees, like in some of those sessions, when you're trying to ask a question to a senior PI or something like that, sometimes that's a little intimidating in person, right? And I was actually doing this morning on some of these sessions, right? There's a Q&A session after the talk. And it's really just a little chat panel, a little square that you can just type your question in. And anybody can do that. Don't worry about getting in front of like 200 people and asking a question, right? So that's very approachable. It kind of levels the playing field a little bit. And I'm a big fan of that.
Yeah, I think that's a huge upside, although I'm going to come back to it in a few minutes on how that's kind of a con. But let me move on. And let me just say, Arun, you better get out of the cardiac concurrence. I have a feeling you're going to be living in those cardiac concurrents. The whole point of this is that you can flit around, all right? So I better see you poking your head around in the pancreas, the endoderm, maybe some hematopoietic. Come on, bro.
Yeah, I'll make it happen. You know me, it's kind of my thing. I love the IPS cardiomyocyte. Tons of good IPS cardiomyocyte stuff here, but yeah, plenty of other stuff going too. So I'm going to keep it below 50%. How about that? I'll try.
Yeah, we'll see. We'll see. Anyway, moving on. Yeah, all those concurrents. I won't belabor you, but Trounson, I work in reproduction now, so Alan Trounson, he's one of the founding fathers by way of the fact that he, derived some of the first ES cell lines from human. He's talking about ovarian cancer. I'm into reproductive biology, so I'm going to get in on that. And Friday, I don't know, Arun, tell me. I want to peek in on the Women in Science panel. Is that am I unwelcome? Probably not, right? I'm sure this is a new era. That I can be a part of, seeing what's moving the field forward for the women, yes?
Yeah, of course. Why not?
Right.
You're of course an ally to other women in science, as am I. So I think that's definitely something I'll stop by.
I mean, I know I'm talking about me specifically. I may be a bit of a wild card. So guys, please let me in. If you see me in there asking questions, don't kick me from the room. And then in the concurrence after that panel, who's run by Christine Mummery, who's the future president, by the way, I got to check in on my boy, Shaheen Rafi, who's got a big talk about this story that I think is about to come out about these kind of versatile endothelial cells. I love endothelium and hematopoiesis. So I'm going to get into that. There's some dendritic cell reprogramming in the same concurrent elsewhere. Actually in the same session, of course. I might be living in the hematopoietic endoderm session, I'm afraid. I should take my own advice. And then, of course, I got to get in on Madeline Lancaster. We're having her on the show. I should also mention that we're going to have Christy Redhorse, right? So I'm going to get on that talk. And unfortunately for my buddy, Justin Achita, who's running parallel to her, you're on the outs, my man. But I know what you're doing anyway, so we'll just have to talk about it over the phone. But a little shout to you, my brother. And then, I don't know about you, but there's this plenary number 5 that takes place my time from 11 to 1:15. And no coincidence, it's about stem cell aging, and there's some kind of vampiric element to it that has it in the nighttime. I don't know if I'm going to make it, Arun. You're on West Coast time. I don't think you have an excuse. Are you going to make it to that?
Yeah, I'm going to try, man. I'm going to try to make it. I mean, I had to wake up at 6 o'clock in the morning to catch some of the morning sessions here today. And, on that note, there's still some really good stuff that's already happened. Some talks that have already been given. There was a couple of workshops on clinical translation. Actually, I think there's going to be 3 of them today. So we were actually both listening to some cool talks about, for example, a heart in a jar coming from Ron Lee at the Karolinska and Nova Heart. That was pretty neat. This 3D organoid-like apparatus where you can actually grow in a jar and you can scale up into high throughput as well. There's some cool talks from Albert Edge and talking about hair cell generation, Georgia Quadrado on organoids. So, this has already happened. This is already going on, and don't be late to the party.
Yeah, there was a lot of talks early, and I have to say, and this is, this is my first virtual conference, so I don't know how it goes at other ones, but this is a big one, right? It's massive, and they're trying to coordinate all these big deal scientists, and it's a bit of a letdown, of course, but I think there's also some growing pains and some adjustments that need to be made. Some of them need to be made on the part of the audience, and some of them, are just technical growing pains. I think you talked about Ron Lee, in that first talk, it was kind of like a bit of a shock because right off the bat, the technical glitches came in, and there was a little bit of loss of signal. And like every time, of course, that the sound cuts out or the video cuts out, the chat room blows up and everyone's spamming the keyboard like, I can't hear, and then everyone freaks out, and it kind of is a bit distracting. But I don't know. I mean, my one piece of advice, and I'm not giving advice to all these august speakers and scientists out there, because I'm not wanting to give advice, but from a technical standpoint, here's my advice. Go to work. Don't do it from your house with your busted cable, modem. All right, go to work with your T3 connection so it can be solid. I mean, for God's sake. And another thing, like, come on, with the screen of the thing, when you're looking down on it, it's looking up your nose, okay? And it's looking at the ceiling and the lights, okay? Here's another thing. When you're sitting in front of a window, I can't see your face because you're all backlit. Okay, so for the audience and the speakers, there's going to be a few things. And for the moderators, Arun, I don't know about you, but I'm looking at these moderators and they're having little mini heart attacks. It looks like it's stressful to be a moderator to begin with, but the faces that I'm seeing on them as they're trying to navigate all the, scaffolding, the questions, and it seems like these people are, I'm not going to try to moderate at one of these virtual conferences ever because it looks intense.
I had no idea that you were such a technical guru, right? Because, you're so active on Twitter and everything. You're the technical expert of the show, aren't you?
Don't sleep on me, Arun. I'm very active on many things technical. I play video games sometimes. I'm a child of this generation of mine. I don't know which one that is, but I have an iPhone, so stop sweating me.
All right, man. I thought you were stuck with one of those little Nokia bricks, but anyways. Speaking of Twitter, though, there's a lot of really awesome stuff that's going on Twitter. Daylon's not going to be able to see this, of course, but if you follow the hashtag ISSCR 2020 hashtag, you can see a live update, live stream of everything that people are talking about, right? I don't actually know if you're allowed to directly tweet fresh data that's coming out of talks on Twitter. I've actually reached out to ISSCR about that, and that'd be good to kind of get some feedback on. But if you want a broad overview of some of the talks that are going on right now, some of the sessions that are happening, and again, another plug for the trainees. A lot of the trainees are advertising that they're giving posters at certain times on Twitter. And so this is a great way to amplify their voices, right? If you're seeing a trainee that you think, hey, this is a really cool poster and I want to go to her talk or go to her poster, and then give that person a retweet or amplify that voice so the trainees can be heard, right? A lot of good stuff going on Twitter, and you got to definitely follow along.
While we're talking about the virtual engagement, I could give you a little brief message from STEMCELL Technologies here. They have their own virtual thing going on, and it's the virtual conference exhibition at ISSCR 2020. It's important for scientists to keep current with the latest advances and innovations, whether you're attending virtually from June 24th to the 27th or just want to see what's new. You can explore this page to stay connected with the field and continue learning and celebrating science. You can browse through the sessions, resources, and upcoming products for your stem cell research. And you can also look at scientific talks and posters that are presented at the ISSCR. You can check that out on the STEMCELL.com website if you search for Virtual Conference Exhibition /ISSCR 2020. Arun, let's get back into the kind of, pros and cons or lessons or takeaways or what do you predict? Let's start with that. What do you think is going to be the major theme for this? Last year it seemed like it was the young investigators, right? That's what they were trying to push. I thought they did a pretty good job about that. We saw a lot of young scientists. Carl Kohler was just on the show as a great example of that. Amazingly, he presented that story there because the emphasis was always on fresh, unpublished research, right? And it was presented there and just published like a couple weeks ago. So that shows how successful that was. There was a lot of novel research there, but I don't know, this time, I don't know what the theme is going to be, but I fear that it can't really be that because you kind of alluded to it there in terms of like retweeting the science. They strictly prohibit video recording and taking pictures, right? And they do that, of course, at the poster sessions and during the conference, when you're on site as well. But then a little bit more easy to regulate, right? Because you see somebody, and even so, you see everybody click, click, click with their phone, taking pictures of the slides. But here, it's pretty much you're just beaming the presentation if you want. We could be recording the whole presentation, disseminating it. So I feel like the investigators this go round are going to be a little bit more conservative on how much they share. I don't know about you.
Yeah, actually, I have the other thought. I think people are going to be pretty open in terms of what they're going to share with everyone because, like you said, this is online. This is already on the internet, as opposed to somebody just snapshotting some pictures of your poster or whatever, right? I think things are going to be very open, and I think this is a positive thing. One thing that a lot of people were wondering was, is this going to be the way for the future? If this goes really well, not just ISSCR, but other major scientific conferences, is this something that we're going to stick with going down the road? This is certainly, of course, you have to worry about time, right? When it comes to the time zones, like right now it's noon in LA, but across the world, it's going to be the dead of midnight. And of course, that's not conducive to listening to science always, right? But if this is another approach to kind of bringing the world together and the scientific world together, I actually think that this is going to carry through, and we're going to see more and more virtual conferences like this. But in terms of like an overall scientific theme, yeah, I think it's tough to decide on one. Of course, we're going to have organoids, we're going to have tissue engineering because they're super hot topics. One thing, intentionally or not, that's going to come up is COVID-19, and we actually have an entire panel on that, and it's going to be a highlight, right? So I think it's tough to say if there's going to be one single scientific focus, but there's a bunch of hot topics to cover. There always are.
That's for sure. I think, I'm going by today, obviously, so I'm biased just because it's the only talks I've seen, but I think there's definitely an emphasis, and there has been in the last few years, a bit of a wave emphasizing the translation, of course, for good reason. So I think we're coming around on that. We got a lot of investigators are talking about how, and not just organoids, because I feel like that's, straightforward and obvious, but like Alison McGeehan in Canada, I forget exactly where, sorry, Alison, my apologies, but she was looking at this Mender system that I thought was really fascinating. It was kind of deconstructing the muscle satellite cell and regenerative processes in vitro, and it was a weird amalgam of the in vitro and in vivo, and like not quite organoid, but like three-dimensional thin sheets, and had an engineering component. So I think that all the science is moving towards this idea of getting up to the physiological scale in terms of function and anatomy of the tissue, but also scaling up in terms of industrial. How do you standardize these protocols and apply them en masse to really get this into a translational sphere? So I can see that theme growing. And you were talking about the things that are going to carry through in terms of just operationally, technically. I think you're right on there, and you talked about it at the outset about the question, the Q&A. Right away, you can see the value of that. As soon as the first session started, part of it is because there's people who are a bit naive to the process who are putting their questions in the chat. And I feel bad for whatever engineers out there have had to correct and write the same text. I'm sure they're just copy pasting, but they're trying to redirect these people to get their Q&A in the right sphere. And I'm sure we'll come around and we'll get it right by the end of the conference. But you could see immediately the impact of that is that they're asking questions that they were too shy to ask before. And on the flip side of that, what you realize is that a lot of the reason that some people ask questions is not really to get the answer as much as to say the words and to be heard and to sound smart. And you could see how people are going crazy and that's why I think some people are putting their questions in the chat, because if you do the Q&A direct, it goes direct to the moderator and the whole chat room doesn't see it. And these people need their question to be seen and have their name attached. Maybe, I don't know, I'm speculating on that. But there's a downside to that too, which is part of the way, as you said right at the outset too, Arun, you're so shrewd, is that these conferences are really a way for the trainees to be recognized and to be visible And downside here is that, I see a bunch of names in the chat room and I see Kevin Egan. And I'm like, okay, Kevin Egan. All right. But everybody else, it's like just a bunch of names, So I hope, like you, that they get some love and that people show up for these poster sessions and we get maybe a little bit more elaborate description and maybe some, pictures of these people so that we can really get them more visibly rendered. For the senior investigators out here who are pretty much losing a year, this is a major point of recruitment for a lot of major investigators out there where they can get the cream of the crop talent. So it's important that we preserve that function of the conference.
Yeah, just a quick note, and we were talking about real-time feedback and just the speed at which, science can be disseminated at this conference. I actually just got a tweet back from the official ISSCR Twitter page saying, "Hi, please do not show unpublished work presented at ISSCR 2020 on your social media channels. Sharing your insights and impressions is great." So there you go, just some clarification for you. Not that that would bother you at all, Daylon, or affect you at all, but just thought you'd know.
No, well, I do. I have, what you guys don't know about me is I have a lurker account for Twitter that I had to, just like this ISSCR interface, I have to wade into the Twitter sphere and understand it first as a spectator before I can show my tweets to the world.
Good to know. You're missing out, man. Maybe that's a contest we can develop. If you can identify Daylon's hidden Twitter account, you get a prize. Maybe not from STEMCELL, maybe from me.
Good luck. Good luck with that, guys. So yeah, that's that. I mean, this is an introductory. We're not going to talk about any specific speakers that we saw today or presentations, but they're good. I mean, I'm optimistic. I don't know about you, Arun, but I'm optimistic about this next few days. I think it's going to be as much of the ISSCR experience as I could hope for in these times. But I mean, I'll just say it's not as fun. Let's be honest. I remember I met you for the first time at last year's ISSCR in LA, and that was a blast. I mean, just the whole idea of meeting, I just remember it fondly, probably because I was actually touching people, but not in an inappropriate way. But I was allowed to have human contact, which I've been a stranger to, in spite of the fact that I'm a married man. Think about that. So I will say, I miss a lot. I miss a lot of the conference, but I think there's still a lot to be gained. Arun, let's have a conclusive take from you, and then we'll wrap.
Yeah, I mean, it's like I said, it's a whole new world. It's gonna be a whole different experience. But speaking just from a trainee's perspective, we still need that exposure. And whether you're doing this in person or whether you're presenting a poster online, this is really critical for your career development. And so I hope that you're, fully invested in this conference as a trainee. Listen to the talks, make connections, talk to people. You can still do that digitally. But also, if you're not a trainee, facilitate the promotion of trainees by retweeting their science or reaching out to them and engaging them. So we can make it as good as we want it to be if we all just kind of work, right?
Some nice parting words, Arun. That brings us to the end of our first ISSCR video episode. Let's hope it's not the last. Well, there's going to be a few more, but maybe we could even renew this. I mean, I'm not so horrified by my own face as I thought I would be. Make sure to follow us on Twitter at STEMCELL Podcast to find out what we're doing at the meeting, and check back here tomorrow for a roundup of all the interesting research that's taken place over the previous 24 hours. We'll see you guys then. Thanks for joining us.